This revision video is going to look at both topics 4 and 5. So it's a big one, but they complement each other. So let's have a look and see. We'll start with some ecology and actually put some evolution throughout it as well. So to begin with, we need to make sure that you're clear on a couple of definitions. They sometimes pop up in exam questions and they expect you to use your language really precisely, particularly in an extended response. So knowing that at a species level, we're looking at organisms that can potentially interbreed, which usually means they'll be living in the same area, so geographically able to meet, and that when they do breed, they produce fertile offspring. So we see things that push the definition sometimes, or things that perhaps wouldn't otherwise come together to produce offspring, like a tiger and a lion, for example. Or the classic example that almost fits the definition is the mule, which is infertile, which is the offspring of a horse and a donkey. So we do see things like that really challenge that definition, and also species that reproduce asexually that doesn't apply to. But for most things, most animals that we'll be looking at, this interbreeding quality helps us to define a species. We can also look at other biochemical evidence, and that's where we tend to look to at evolutionary history of organisms. Population is sort of moving up the scale here, where we've got a group of organisms of the same species. So they would be interacting and potentially interbreeding and being a sort of a defined geographic area. So you might have, a population of, say here we've got this sort of antelope in one particular grassland, or a forest or a mountain range or something you might have. The community then extends to lots of different species that are all interacting. So all of the biotic components and anything that's alive, so that includes bacteria as well that could be in the soil, for example. An ecosystem goes to both biotic and abiotic. So even the rocks, the water, the air, the clay, minerals as well. A little refresher as well before we go any further is just to remember the equation for respiration and photosynthesis. So here, this is our glucose, or an example of a carbohydrate, combining with oxygen gas to make carbon dioxide and water. And we're going that way, we're talking about respiration. And we get an energy output that could be used for things like movement, creating sound even, and ultimately it's all going to become heat. And then back the other way, we have an input of energy in the form of light that helps drive the creation of this larger organic molecule. So this side, we have something is organic and energy rich, and this side, everything is inorganic. And that's important because that's how we define autotrophs and heterotrophs based on whether they obtain organic or inorganic substances from the environment. So an autotroph means self-feeding. The troph part means feeding, and auto meaning self. Most of the things we deal with are these, the photoautotrophs, so carrying out photosynthesis, particularly in terrestrial ecosystems, which means on land. And even some of the aquatic ones or marine, we're mostly limited to the phototrophic kind of zone that where there's enough light. But there are things to a small degree, these chemoautotrophs that live in sort of deep sea volcanic vents and other places where light isn't available, and they've found an alternative way to obtain energy. But something like this here is still an inorganic molecule. On the other hand, things like ourselves, we are heterotrophs. We cannot go outside and photosynthesize. So we are a consumer. When I say we eat living organisms or recently dead, or recently killed, and that can include plants and animals. Killed sounds a bit vicious, but it could just mean, a vegetable taken off the bush. So we fall into the category of consumers. You can split that further. For example, primary consumers will eat vegetation, secondary consumers will eat the primary consumers, and so on. Or some organisms are adapted to survive off things that are dead and decaying already. And we break them into two groups. The detritivores are the ones that eat their food first. They ingest it, they take it into their bodies, Another way to say that is that their digestion is internal, so like in a stomach or a gut, whereas saprotrophs are the ones that digest things before they absorb the products. So that's also known as external digestion. If you're having trouble distinguishing between those, you remember there's a difference, but you can't remember which is which. This vor part here is from Latin, from vorare to devour. And the same as like carnivore, herbivore, something that actually does a like sort of physical process of eating, if that helps you. There are also things that don't fit the category perfectly, like a Venus flytrap here. We would still consider that, for the most part, autotrophic. That's its dominant sort of mode of nutrition because it's photosynthesizing to get energy. It's just supplementing, it's living in sort of nutrient poor soils, and it's supplementing that its inorganic nutrients by trapping insects. But we'd still consider that an autotroph. When we look at then ecosystems and the role of producers and consumers, energy moves through an ecosystem in a single direction. So it enters by light and ultimately it all leaves as heat, because all energy transformations will ultimately result in the creation of heat. Whereas matter has to be recycled. We have lots of nutrient cycles, and the carbon one is the one we focus on in our course, but also nitrogen, phosphorus, everything else, any other part of matter has to be cycled around as well. Or you've probably seen the water cycle, perhaps in geography too. So those things, those saprotrophs, or we might also call them decomposers, is another word you would have seen. They help those cycles. Without them, we wouldn't have a lot of things returned to the soil, for example, for new growth. Now, when we look at diagrams of these sorts of things or calculating losses, if our energy comes in as light, and then we're saying most of it leaves as heat, so you might have some movement, you might have sound even, or new energy-rich chemicals that store that energy that's come from light. But ultimately, as it gets broken down and transformed, it's tending towards heat, which, for anyone who's interested, looking at sort of second law of thermodynamics there, that in any isolated system there's a tendency towards entropy, so energy becomes more disordered. That's maybe a bit fancy for what we need, but in this context, it just means that in an ecosystem, all the energy that comes in ultimately becomes heat. And it doesn't disappear, or that would break the first law of thermodynamics, because energy can't be created or destroyed, but it does dissipate, it does spread out. That heat through our atmosphere, and then we have losses to space. Now, some implications of that: if we can't get energy or magically make it appear, and we're losing lots all the way, and it's approximately 90% with each trophic level, so with each step in a food chain, there's a limit. We can't just have food chains going on forever, because you'd need an enormous number of producers to support one or two top carnivores here, big predators. So there is a limitation to the length for those, and it also means that the energy available at these top trophic levels is much, much, much less than at the bottom. Or another visual to see that, if we looked at all that light energy absorbed by plants, by the time we get to the top of that food chain, there's very little left. But all of that ultimately, you can see heat, heat, heat. That's where it's going. And aside from having limited energy available at the top trophic levels, you also have losses of biomass. So we don't have huge stores, like you can't have a greater mass of predators essentially than you have of your producers. And we get losses of carbon dioxide and water and other waste like urea as well. And even if you think about it, as well as just the energy transformations between each trophic level, not everything gets eaten. So some of these pea plants here, for example, the aphids are not going to eat the roots. And we get up to here, perhaps like a robin, a hawk might eat the robin, but it doesn't digest the boat. They will get spit back up, and it won't be part of the energy source for the hawk. We can represent this as well using a pyramid of energy. So this pyramid here, but it's in stepped boxes. So please don't draw something that's sort of triangular. Don't do that. It needs to be boxes, and it's really kind of like a bar graph. It's just sort of mirror image, like a bar graph that's been centered in a Word document. I can just show you as well here too, like this kind of terminology is preferred by the IB, talking about a producer, something that produces the organic components, so our photoautotrophs here. And then the things that eat them are the primary consumers, so the first consumers in the food chain, and then you move up. Now, of course, these sorts of things are quite simplified. Like this is only showing you a food chain, which is where there is a single link, so one organism at each level. But in reality, food webs and ecosystems are a lot more complex, kind of tangled up. Like here's an Arctic one, for example. And you might be asked, you might have something like this, probably without pictures for your exam, but it might name things or it might even just give you like letters or numbers. And you would have to identify, for example, that this is a producer because there are no arrows going into it. So even if you didn't know that phytoplankton did photosynthesis, you would know that's a producer. And the arrows, we can see little heads of those arrows, that's a direction of energy movement. So they have to be drawn that way. And we can see that krill, for example, is a primary consumer because that is the first thing to eat phytoplankton. But the krill also eats some of the other zooplankton. So if we looked at that food chain and we went 1, 2, 3, it's actually the secondary consumer. It's the next thing to eat. The producer has gone via something else first. So the context will be specified for you, or you might just get a simplification to work out a question or as an example. Now, talking about energy and matter moving through ecosystems, one of the key things for matter is carbon. Here's an example where the arrows are sort of roughly proportional to the size of the flux. So a flux is a movement in between each of the sinks. A sink is sort of another name for a store. And that's also, in this particular diagram, they've tried to make the boxes roughly proportional as well for the size of the sink. So try to use that terminology, a sink for where you find the carbon and a flux for the process of being exchanged in between them. So we can see here one of the biggest things happening is carbon going from the atmosphere into plants. So photosynthesis is the biggest flux. Now, some of the information will be a bit variable, but we can see that particularly even just in the water as bicarbonate ions or hydrogen carbonate here, but also in animals and even detritus in marine ecosystems, mostly by sheer size, the volume of water, that's actually our biggest sink of carbon. And we're really altering that with climate change at the moment, unfortunately. We're also really altering this flux here. Because ordinarily, we would not expect carbon stored way down low in the ground to be combusted in the atmosphere. So we're having a really big impact on the carbon cycle. Here's another version of that diagram that's just been a bit simplified. And I just want to point out that if you are doing something like this for an exam, for an IB exam, you should be able to construct a diagram similar to this. You won't have to include necessarily every possibility. There'll be a set number of marks, but perhaps one mark might be worth 2 components on your diagram. So it's always good, or as a little insurance policy, to try and think of a few extra things to make sure you get the marks. But you can see here, this one, it's just talking about organic compounds. So you don't need to name glucose or amino acids or anything like that. And likewise, carbon dioxide in the atmosphere, okay, that name's there, but if you just said carbon, that'd be okay if you wrote CO2. You can talk about carbon in fossil fuels. You don't need to say that they're hydrocarbons or even, things in water. If you just said carbon or inorganic carbon would be another thing you could say for things like CO2 or that HCO3. If you're including that in the water, you could just say that was inorganic. There's the emphasis taken away from the naming because that's really the realm of chemistry. These processes, however, you should be able to say that respiration is what's happening, and that's occurring in plants and animals. Don't forget that. It's a common thing to forget that plants respire. And of course, photosynthesis and combustion, as we mentioned before, that's burning. This term fossilization is a bit of a strange one. Or sometimes they use sedimentation, but they're talking here about the creation of fossil fuels. And then even things like just feeding and death, you can use sort of everyday terms for those. Now, if we go back to our marine things for a moment, when we look at sort of where some of that carbon is stored in the ocean, apart from being dissolved in water, we actually see that a lot of organisms that live in the sea use calcium carbonate as part of their bodies. So here, these shells, for example, are made of calcium carbonate, or in corals as well, or processes of sedimentation give us limestone that's also made of those shells over a long period of time that's become rock. Now, part of this course also looks at climate change, and one of the big problems at the moment is ocean acidification. So by putting more carbon dioxide into the atmosphere, it also means more of it's getting dissolved in the oceans, in water, and that's generating more acid, carbonic acid. And what that will do will actually turn carbonate ions into bicarbonate ions. And again, the emphasis isn't on the chemistry, but if you know that it just means that there's less carbonate or less carbon available for those organisms to make their shells, make those rigid structures in corals, for example, in coral reefs, then you can see why that's a big problem for those ecosystems. It also means too that if the water is more acidic, the existing structures are going to get more easily dissolved. All right, if we turn back to a few other things that can happen to carbon, when organisms die, of course, we generally expect them to get completely broken down. That's the process of respiration then by those decomposers, things like our saprotrophs. And of course, they're going to return carbon dioxide to the air or water to continue in the carbon cycle. But another option that's possible, some of these archaeans, so if you've forgotten that part of classification, we're in the domain eukaryote. Eukaryote are here, and bacteria or prokaryotes are here, and the archaea are here, another branch. So they're a very, very ancient lineage of single-celled organisms, those ones that live in those extreme environments like highly acidic pools and thermal pools and things, high temperatures, our extremophiles. So some groups of archaeans actually produce methane instead of carbon dioxide. So that's an alternate pathway for carbon that we see in the environment. Now, for those the conditions required, they have to be anaerobic. Anaerobic means no oxygen, which is why the carbon, see here in carbon dioxide, carbon is paired up with oxygen. It isn't paired up with oxygen in an anaerobic condition. So sometimes that could happen, in sort of swampy kind of areas where you've got like waterlogged soils, for example. And oxygen can't get through. So that's where we might be getting like some of that methane. And methane is actually a very potent greenhouse gas. It's very good at trapping heat in the atmosphere, even though there's actually not that much of it. Another thing that can happen though too is if the conditions are too acidic for the particular archaea that are methanogenic, that make methane. So for those ones that aren't adapted to those conditions and the pH drops too low, that's when we don't get the full decomposition. So instead of that carbon breaking down and all becoming methane, we get larger molecules that aren't as completely broken down, and they tend to turn to our fossil fuels. Although actually gas is the methane, but a combination, these are bigger, the coal and oil. They're larger, less broken down forms of organic compounds. They still need to be in anaerobic conditions though, because what you get, fossil fuels are long chains of hydrocarbons, all sorts of lengths. I won't draw so on and so forth, but there's no oxygen in them. So they would get broken down. If there was oxygen, they would break down as carbon dioxide. But for this to happen, a lot of high temperatures and a lot of pressure is needed to sort of compact them and crush them down. And so they're only found in certain environments where there are porous rocks and over a really long period of time. So if we went back to that carbon cycle, we'd be thinking, well, actually the flux, the movement of things into that sink of fossil fuels is really slow. So relative to that, the rate at which we are extracting them and combusting them is very disproportionate. And we can see that that's what we're doing. Which takes us to talking about the greenhouse effect for a little bit. Now, what that is, if you're not familiar, given first of all, is like a structure made of glass that you put plants in, because greenhouse with its glass lets light in, but once things warm up inside, the warm air gets trapped, and so it's warmer inside the greenhouse than out. Now, that's quite a good thing that we have that same kind of effect happening on Earth. So sunlight comes in here, it gets absorbed by plants, the soil, animals, lots of structures, and so they warm up and re-radiate infrared light instead. So that's here. Now that relates to this graph over here. So this is visible light on our electromagnetic spectrum. So that's a lot of the incoming radiation from the sun. And then what the Earth tends to emit back is longer wavelength here, in this infrared zone. So it's not quite the same quality of light, and it's not visible. It's not light that we can see, but it's that kind of thing that if you go out on a hot road, hot day in summer, and you're standing on a road that's black, that's absorbing a lot of visible light, if you hold your hand, if you hover it just above and you're not touching it, you'll be able to feel that radiating heat coming back off it. Now, the thing is with these greenhouse gases, the main ones are carbon dioxide and water vapor, and they do absolutely exist naturally. And it's a really good thing that they actually absorb some of that infrared radiation. We can also refer to that as short and long wave, the short being the visible and the long being the infrared. So they're good at absorbing that long wave radiation. Let me just write that. And that's so short. Some people sort of talk about thickening the atmosphere or a blanket, and that's not quite. I can see where they're going with the analogy, but it's not that we're actually making the gases like a high pressure or anything, but we're changing the composition of gases. So what would normally keep in a bit of heat for our atmosphere and sort of help keep our temperatures steady and suitable for life, we're changing the proportions so that we're trapping more heat than we used to. And that's why our global temperatures are on the rise. And of course, when we talk about climate change and global warming, it's warming on average. Some areas are actually getting cooler because we're also changing weather patterns and circulation of those sort of hotter air currents and water currents. And then we mentioned that methane is one that's quite effective, although we don't have that much of it. And also, you don't have to worry so much, but nitrogen oxides are another, probably the fourth on the list for greenhouse gases and their contribution. And we can see here, for example, that like carbon dioxide falls in this kind of region of some of our re-radiated light from Earth. So it's able to absorb some of that energy and keep it trapped here, keep radiating it, sending it back down. It's just scattering it, but it's preventing it from being lost out to space. One of the things that you can be asked about, that's popped up on exams, is this graph here. Now, Mauna Loa is an observatory in Hawaii where they do a lot of testing. I think it's the highest active volcano in the world, and they do a lot of testing, sending balloons up to sort of calculate and measure the parts per million, the proportion of carbon dioxide in the atmosphere. And we can see here that year to year it goes up and down. And that's actually just got to do with the fact that we put the equator on there. We can see that most of the landmass is in fact in the northern hemisphere. So their spring-summer season brings the global carbon dioxide down, and then our spring-summer in the southern hemisphere isn't enough to offset it, so that it goes back up again in their autumn and winter here. Also, you find that we don't have as much land mass in sort of colder climates. So you can see here, we don't have or quite the range of what we've got, down here with Antarctica at the bottom. Don't forget too that that's an icy desert. So we don't have any sort of photosynthesis happening here. Now, how we might study some of these other things, and sort of coming back to the ecology in the earlier topics, is the idea of a mesocosm. So some of the things that we're doing are whole enclosed sample ecosystems, like this biosphere project over here. Or we might be doing things, for example, isolating sort of columns of water and making little tanks, sealing them up and changing the carbon dioxide and actually seeing what that's doing to how whole food webs are responding, rates of photosynthesis and things like that. Often for those sorts of things, if we're wanting to test something like climate change, we'll use a sealed mesocosm. That's considered closed. Closed means it allows energy in and out, but not matter. Whereas open is open to both. So that means like air and water can come in and out particularly. But the concept of a mesocosm is quite broad. This means an isolated, or not completely isolated, sorry, but sort of a separated little study area for research that kind of replicates things that might be happening in an ecosystem. Another thing we might do when we're measuring in ecology, quadrat sampling is quite a popular thing for testing things that don't run away. So it could be vegetation or it could be sessile animals, things that don't move. This picture here shows a transit line, which means you might be investigating a gradient from one end to the other. Or you could be using this quadrat frame here and distributing it randomly to sort of take some data. Could be another way you're doing it. That, by the way, I'll just write quadrat. Spelled differently to quadrant that your autocorrect or Microsoft Word spell check will probably try to change it to. Quadrant is not the same. So let's say they were random instead. Here we've got some Neptune's necklace, this seaweed, and a rib top shell, this little sea snail here. And we might say, for example, in a study of fish, we tried to see which species were present in either. So we might have some, just the one species present in some, and some where they overlap and some with neither. So we'd have to set up a frequency table here, and so we can see where we've got both or neither. I'm just going to fill them in and go through that fairly quickly, if I can. I'm getting things saying my network quality is poor, so I hope this is still recording. But just putting that information in, and then the totals for each row here going across, and then each column as well. So 28 and 7 is 35, and then 35 and 15 here equal 50, the total number of quadrats, and these two should equal 50 as well if you've done it correctly. All right, keep that table there. We then look at sort of writing our hypothesis, and a null hypothesis is when there's no significant correlation here, no statistical significance. So we're trying to work out what's going on. So that's reference, that little text is reference to another way you can use chi-square, but let me skim past that for a moment. All right, so what we can see here, we've got our total number of quadrats with the snails, total number of quadrats with the seaweed, and so on. If we multiply these together, then that tells us how many we should expect to find both in, if depending on if there's any significant overlap. So if I just highlight those, for example, here, if I thought theoretically, I was going to like what sort of a neutral result where it's just by like randomly due to chance. 35 with snails, 37 with seaweed. In theory, if I multiply them together and then divide them by the 50 quadrats that I use, because there are 50 in total, then in theory I should get only 25.9 quadrats where they're both present. So I need to do that for every possible, like all these 4 squares here. I need to take the number from the end of the row and the bottom of the column, multiply them together and divide by total quadrats. So I can fill in the rest then. So that's 13 times 35 over 50 is 9.1. 15 times 37 over 50 is 11.1. And 15 times 13 over 50 is 3.9. So they would be the expected number of each quadrat. What if you took another 50 and there was no overlap, no significant correlation between the two species, if it was just by chance which ones you found them in, based on just how frequent they are. Now, to do the calculation, I've kind of tried to sort of set it up in stages, because the formula here is this bit. It's the sum of all the differences between what you observed, the numbers in black, and what you might expect, the numbers in red, squared, and then divided by the expected number again. So it looks a bit messy, but they're probably not going to give you a full calculation to do in an exam. There's going to be something that applies the result, is more likely. So here, observed minus expected, squared, and then they often actually work out to be the same. And then, but what changes is the last column. When you do this, observed minus expected squared and divided by the expected value again. So for Neptune's necklace and rib top shells, that's, sorry, I've circled the wrong thing there. That's our expected value there. So when I add those all together, because we want that sum of all of those at the end, and my stylus is so good, we get this result here, chi-squared value of 2.18. Now that's when you have to look at these sort of probability tables. And the accepted one we use is a probability of 0.05, well, that's the same as saying 5%. So that's what we have a look at, say a 5% chance or 5% probability that it's just random, whether or not we're over that limit or not. So what we calculated, 2.18 here, that's not greater than the critical value on our table. So that means we can't reject the null hypothesis. So it could just be due to chance that these species sometimes are found together. It could just be because they're both just so prevalent on the rocky shore. So we can't say that we're sure there's a correlation there. If, however, it was higher than that number, we'd say, oh, statistically, there's a significant pattern here that they appear together more frequently than they would if it was just random. Now, when you choose here, how to know which row to read in your table is actually comes from the previous information here, this table. How many rows are there and how many columns of the original data? That's what we're looking at here, not the totals at the end. So here we've got 2 rows and we've got 2 columns. And when you work out degrees of freedom, this is sometimes a multi-choice question in paper 1. Which would you use from a table like this? It's however many rows you've got takeaway 1. So we had 2 rows. And however many columns, 2 of those, let's say takeaway 1, apologies. So it's just 2 minus 1 is 1, 2 minus 1 is 1. So 1 times 1 is 1. That's how we knew the degrees of freedom. You can ignore that, sorry, this is from another example, but the table is the same. All right, let's now look at some things to do with evolution. So evolution is cumulative change in heritable characteristics. It's usually specifically in a particular population. Now, when we talk about heritable characteristics, we're talking about genes and alleles. Sometimes even whole chromosomes. But usually, most frequently, we're looking at a change in allele level. So different variations of the same gene and then becoming increasingly frequent, a particular allele in a population. So that cannot happen to a single organism. One organism cannot evolve. The population can, because you have to have subsequent generations to be able to pass things on and to see change. Now, the process, we talk about evolution occurring by the process of natural selection. Now, typically, we would need to see some reason for competition. So we call this overproduction, or telling us that there are more offspring, more babies, than can be supported by the environment, which causes them to compete for resources. Now, there has to be some variation already. Now, this is existing variation. Variation isn't created by natural selection. Variation is already there due to chance mutations. So there's slight differences in the population. That means some of them are already better adapted to survive. And that's the selection part. Natural selection means the environment or nature. So it could be resources or a predator or whatever, various abiotic and biotic factors, but in the environment are selecting better opportunities, I suppose, for certain individuals to survive. So the ones that are best adapted are the ones that survive and they can reproduce. So their offspring, if it's something heritable, if their offspring will have the alleles or perhaps a new gene or even a whole new chromosome, but most commonly the alleles of the parents that were successful. And that means in the new population, the proportion of that allele, so let's say it was for this darker coat colour, the proportion of that allele, the frequency at which it appears in the population increases. And that's the increase in frequency is the evolution bit. And the killing off, if you like, of the less fit for survival is the natural selection part. So we talk about them together as how evolution occurs by natural selection. Now, the mutations that we talked about in the variation, so that produces new alleles or sometimes even whole new like gene fragments or other products. And also in species that sexually reproduce, meiosis will create new combinations, and the process of sexual reproduction means that you'll get alleles from both parents. So that helps create more variety. That means too that species that reproduce asexually are reliant only on mutation. So they perhaps can't respond quite as quickly to environmental change in that respect, although a lot of them have much, much, much shorter generation times. So that's often to their advantage. For example, things like bacteria. Now, the evidence that species have arisen via evolution comes from particularly from fossil records. So we can see gradual changes in some species. We can see ancestral forms of things that exist today, and we can see links in the time frame. And also, it tends to match the complexity we would expect. So that the oldest fossils are the simpler organisms, structurally simpler, I should say. We do tend to, as humans, put ourselves at the top of the hierarchy, which is quite an egotistic thing to do. Other evidence comes from sort of morphological things. So homologous structures are the things that we share that have a common ancestry and common internal structure. The classic being this pentadactyl limb. So here, these bones are the same. They've changed in shape a bit and have become adapted for different purposes, but we have the same sort of pieces, if you like, as lots of other mammals here, as we can see in this diagram. So that's a good example that we've diverged. So we've gone different pathways. So to diverge means to branch off, or adaptive radiation. Radiation means going out sort of like in a circle. So we've adapted out in different ways from something that was common in the centre. So there's some terms that we might use with that. Another thing is vestigial. Vestigial means a leftover or left behind. So the fact that we have an appendix where perhaps has limited use, people can get by without it, perhaps has a small role to play, but it's certainly not like the big cecum, the much, much larger version we see in other herbivores. It could be a bit of an evolutionary leftover in us. Or we see here, for example, these pelvic bones in some whale species that don't appear to have any particular function, but give us a clue that perhaps that was once a land-dwelling organism that then moved into the sea. Some of those structures it mostly got rid of because they would have been in the way and disadvantageous for survival. But these little vestiges here that remain don't really affect survival and don't really affect their natural selection. So they've remained because there's been no reason to get rid of them. Another thing is selective breeding, which is an artificial way of sort of doing our own natural selection as humans, stepping in and playing that role with the environment by choosing which species to breed together. So it shows us the power of reproductively exaggerating traits and concentrating characteristics and getting down to sort of pure breeding of alleles and making the most of the variation that exists already. So we're able to do things like dog breeds, and also here we can see domestic corn from its ancestors greatly changed by constantly selecting for the ones with larger kernels and sweeter, juicier vegetable and things like that. So even though that's not the same as natural selection, because it's not pressure from the environment, it's the concept of those alleles, the frequency changing over time. Now, if we look at some other examples, see here, this one, this is our industrial melanism. Let's just make sure. Because melanin is a pigment. This is a little moth over here that exists on this lichen-covered tree. And then when we had industrial revolution and lots more pollution and sooty deposits on the trees that killed off the lichens particularly, it wasn't that it just coated the trees in soot, but a bit of that, but mostly killing off the lichens, that the darker variety then had a better camouflage. And that swung back the other way with Clean Air Acts and a change in our industry and sort of restrictions on pollution and our practices. But during those times we can see, you can imagine if you're a predator of that moth, which one is going to be easier to pick off. So that's a selection pressure that's changed in different directions over time. And here's another example over here of that adaptive radiation that from a common ancestor, from a finch in the Galapagos Islands, that birds that arrived on different islands were presented with different environmental pressures for possible food sources. So the natural selection has occurred, that from the variation in that original finch and as well some successive mutations over time, the ones that had advantageous features that were better adapted were the ones that could pass on those alleles, and they became more frequent in each population. And actually when we talk about classification and speciation, it's a process of making new species that are no longer able to interbreed if they come together. And that's what happened to some of these. They were reproductively isolated for a long time on separate islands. So geographical barriers preventing them from coming together meant that they became not only distinct populations, but over a long period of time, all those differences at a genetic and biochemical level mean that when they come together, they don't interbreed successfully. Now, that's not true of all of them. Some of them are still close enough, and we can see some hybridization happening with some of them as well. One thing to be wary of when we look at some of these structures and talking about evidence for evolution, some things can be a bit misleading at the surface. Like here's an example here of what some Australian animals look like and some counterparts from the Northern Hemisphere particularly. Things like sort of body shape or spots and stripes and modes of eating and ecological niche, so sort of their way of living and where they live. Now these have they're mammals, but they have much more distantly related ancestors here, or a common ancestor from much, much longer ago. And these adaptations have arisen separately in these animals after they've already split off from each other. So they're considered convergent, which is when things come in towards each other, rather than divergent, which is where they went out. So that's convergent. So they become more similar over time, but that doesn't mean that they have an evolutionary ancestor recently. So sometimes it's better to look at things biochemically. So here, running these kind of gene sequences and looking for how many differences and similarities. Now, you don't have to do that with a whole genome. You might focus on something like cytochrome C, which is used in the pathway of aerobic respiration. And we can see here then that gives us a good kind of map. And we'll call this a cladogram. So it's a little kind of branch tree that shows us where these connections, these nodes, where we think they branched off from each other based on the cumulative number of differences. Here's another one. This one's a little bit more simplified, and the kind of thing in an exam you'll perhaps have to draw just simple straight lines that come off from one another here. This one tells us changes in beta hemoglobin. So again, if we look at perhaps organisms that all have that molecule, how they're different. And we can do this because all organisms are using the same amino acids, but there will be small differences that have occurred with mutations over time. One of the things, and there's some danger in doing this too, but for the most part, it seems to hold true. Apart from seeing how related things are to each other, the number of differences, we can mostly assume that more differences give us a bit of a molecular clock. So it gives us a bit of a timeline for when things changed. So here, this one's for nucleotide substitution. So this is at a genetic level, and seeing changes that have occurred seem to match pretty well with sort of the arrival of different species and when they sort of branched off that family tree, if you like. Here's another way you might see cladograms. So rather than being kind of a line with branches, I might do it the other way, sorry, that one is so for like coming off like that. We might see them here, kind of like square sort of branches, that's fine too. But we can see here that this looks at people and that saying that Japanese people and European people are split off from each other as populations of humans more recently than they did with African people. And then these animals, if we go back to a species level, that, for example, orangutans here are more distantly related to people than gorillas are, and both of them less so than chimpanzees. But of course, we must have some care doing things like this, because the way we write these, there's an inherent kind of danger that we could put whoever we put as the most recent branch or at the top is sort of considered most evolved, and that's really quite dangerous when we're talking about differences between human populations, and also just thinking more kind of philosophically about where we place ourselves in the animal kingdom, because how recently we appeared in evolution or branched off from something does not mean that we are more or less adapted to our environment than anything else. If we want to make our own cladogram, we would look for how many similarities and differences there are between the organisms that are presented to us. So this is using kind of like binary code, or you could use ticks and crosses to see that, you know, the number of common features, if we went through this list, having a backbone is common to everything, but having like an amniotic egg is only common to lizards, bears, chimps, and humans. Until we get down and we can see which things, so lampreys and humans are by far the most different on this list, and our closest relative here is the chimp. And so we would draw a line, and then we would place those organisms in order and connect them to that original line. And we can see at this node here, this branch, that that's where we must have had a common ancestor with a that was vertebrate. Notice too, even though this order is arbitrary, and again, that's that thing, that warning of saying, well, we put ourselves at the end because we kind of think of ourselves as the best or most evolved or advanced. But actually what all this tells us chimps and humans are different from each other, but it doesn't tell us which one is more like the bear. Both of us branched off at the same point from the bear. And again, similarly here, this sunfish is equally related to a bear or a chimpanzee because we have the same number of these nodes in between. Now, some of the things with cladistics, we've actually been able to do some new classifications and using biochemical evidence. So lots of things that used to be put into the figwort family, so a lot of plants that look a bit like this, and now broken into lots of other different classifications, and some of them quite just. So it's that thing too of that convergent evolution, that some of those species might look quite similar superficially, but if we look at the biochemical evidence, it's likely that some of them broke off much longer ago. So classification now is trying to keep up with sort of phylogeny, that tree of life based on the more biochemical evidence. Classically, we would have looked for morphological, like structural features. Also, even the way we classify birds and dinosaurs, we now kind of think perhaps some dinosaurs would technically be classified as birds rather than some of them were the precursors to. Or we think perhaps even the birds we know now, they split off at a different point in time to what we originally thought and where sort of that warm-bloodedness occurred, which is called endothermy, by the way. Now, looking at cladistics, some of the strengths are that it's quite objective using that biochemistry rather than the morphological stuff, because we tend to look at which features put sort of attract our eye, what we think is more important when we compare structures. So it's less subjective, but it is based on that assumption that the rate of change of like mutations is reliable. It's kind of a clock back through time, or even there might be a change, there might be a mutation that separates some species and makes them more different. And then by chance, one of the mutations could actually revert it back to what it used to be. That is possible. So we do make some, the number of differences between species means they must be close. So to make it stronger, you should try to use several different biochemical evidence, not just rely on one molecule. Now, more to our classic sort of classification then, or why it's useful, why we would want to know which species are most closely related. There's some predictive value to that, or understanding that evolutionary pathway helps us sort of be introspective about ourselves as well. It also helps us identify species and be able to talk about them and know we're talking about the same thing when we talk to somebody perhaps on the other side of the world, or to be able to care for ecosystems better or understand sort of how we can protect species as well. When we name them, we have a two-name naming system. You would be familiar with that. Write it in italics or underline it by hand. And once you've used it once, you can shorten the genus name. That's genus, by the way, the first one, and the species name is the second. So the words generic and specific can help you remember that. The kingdoms. We look at sort of a 5-kingdom system, but actually we now kind of change how we look at it. We tend to look at a 3-domain system is where we're at the moment, most biologists, and then we break into kingdoms within each one. And we're focusing today on the kingdoms within the eukaryotes, which includes ourselves. But these are the 3 domains you should know: the eubacteria, the true bacteria, eukaryotes, true nucleus, and the archaea, the ancient ones. You also need to know some examples or how we classify. So under domain, so obviously that's the domain Eukaryota, and then kingdom, so for us, Animalia, and then underneath that is the phylum, Chordata, class, Mammalia, order, primates, family, Hominidae, genus Homo, and species Sapiens. Or here's another example. You kind of see it as a bit of a diagram, like a group within a group. These ones here don't need to be italicized, only the genus and species. If you're not sure which level of classification is, you can call it just a clade, or the other word you might see is taxonomy. Some other examples to explore at your own leisure. You should know one plant one and one animal one. Try and pick something that perhaps is memorable for you, species you're familiar with, or names that are easy for you to jog your memory. Like here, for example, you might want to memorize the chimp because most of it's the same as us. And then pan troglodytes isn't too bad. You might have heard of it before. And just being okay, and then here we talk about sort of branches of classification, and we modify them a lot depending on sort of that biochemistry and the cladistics that we talked about. But some key things we need to know. If we focus in, so these are our domains, and then here's our kingdom for plants. If we look at key with students, or no roots or stems, like they just have rhizoids, these kind of little hairs that come off them, and very, very simple little spores. So it's not a true stem. They have something quite simple, but it's not the same as a vascularized one. They don't survive without sort of a constant supply of water, so they don't have any adaptations to living in dry environments. Our felicinophytes are our ferns, easy, F and F, remember those ones? They do have vascular tissues. Everything from this point forward is vascularized. So little, like, when you see the stringy bits in celery, so little tubes that carry water and sap through the plant. These ones make spores, these little sporangia, like little dots under the leaf. But they're quite, they don't have any seeds, and they certainly don't have flowers or fruit. So they're still at the level of making spores for their reproduction. It's a good way to distinguish them. Conifera are our sort of pine cones and things like that. You think cones, conifers. So they're very reduced leaves. So we tend to call them needles, like pine needles. They are, again, saying everything's vascularized from this point on, but these ones make seeds. So they have cones that have their pollen or ovules. So male and female cones, sometimes they're on the same tree. And they're sort of woody stemmed big trees as well. Hopefully you'll be able to identify one mostly just by its pine needles or pine cones alone, but we're looking at that information of having vascular tissue and having that, not spores anymore, having pollen and ovules, but having them containing cones. And then our angiosperms are the ones that produce flowers. So they have quite a distinct reproductive system there. And once the ovules are fertilized and become seeds that develop there in the ovary, they'll get a fruit around them as well. So the seed is actually already fertilized. That's got it ready to become a new plant, and it's got several cells in there. It's not just a zygote, it's more than that. And it'll have a fruit develop around it, and that's from a fertilized flower. All right, if we look at some of the phyla within that we need to know. You need to know porifera. If you think pores maybe for holes, sponges, these have no symmetry. They are just really just a blob of cells. They don't have a lot of specialized systems. They have some. I feel like maybe I'm being a bit unfair to the poor sponges, but they don't have, for example, a nervous system. There's certainly no circulatory system. They just sort of have an opening here for little bits of small microscopic bits of food to come in. Some of them you can actually separate the cells through a fine sieve and they will reform on the other side, which is quite amazing. Cnidaria are our next ones, and these ones have radial symmetry, so like here, both ways here going across. They do have a gut, but it only has one opening, so it'll take things in through kind of a approximate mouth. It's not much of a mouth, but things that they don't need kind of get ejected back out. Most of them have stinging cells. So if you think of jellyfish, and some of them, or they might alternate, but they have some sessile stages, which means they're still, and motile means moving. Moving on from there, and this is through evolutionary time, by the way, that we're going with these. The platyhelminths are our flatworms. Platy means flat. So you might think too like a platypus has a flat bill. They also known as coelomates, like they don't have a coelom at this point, but they have and no circulatory system, there's no blood, but they do actually have a gut, a little bit more elaborate, but it's still only got just sort of a mouth, and they're kind of spitting out the things they don't need. They're hermaphroditic as well. Some of them look pretty cool, but they're definitely flat, and they need to be, because if you think back to that surface area to volume ratios of diffusion, they have to get things like oxygen into their bodies without any blood to move it around. Annelids, on the other hand, do have, and they're segmented, and they include things like our earthworms and our lovely leech here. I should have pointed out too that everything then from platyhelminths have bilateral symmetry, which means only one line of symmetry, so not across anymore. That wouldn't be right. One ends, they've got a front and a back. They don't have a skeleton, but that's made of bones, but they do use pressure to push against, so a hydroskeleton. And they do have some gas exchange that can be quite simple, like a little gill system, and they have blood to be able to circulate oxygen around their body. And now their gut actually just goes one way. Mollusks, moving up again, a lot of them have shells or some sort of structure or feeding structure like a radula, which is like a rough tooth-like tongue for scraping the rocks, like this little limpet does here. They include things like gastropods, gastro for stomach, pod for foot, the foot on the stomach, cephalopods or cephalopods if you're going for the Greek, cephalos, so that means their foot on their head, like our octopus, bivalves like our mollusk here. So these ones do have sort of quite well-developed circulation with gills or lungs, and a lot of them have shells too. So it's a bit broad. There's no kind of one hard and fast thing for our mollusks here. Our arthropods have segmented bodies, however. That is something that is quite distinctive for them. And that exoskeleton, a skeleton on the outside made of chitin. And that they will struggle when that gets a bit tight for them. They actually have to molt, get rid of it, and they'll be quite vulnerable until they harden the new skeleton that's starting to grow underneath. They have little jointed limbs, and they include things like our insects, but also our arachnids, like our spiders, and crustaceans like our crabs. And then we get to chordata, which is a huge group, including us. And it's not quite synonymous with vertebrate, but it almost is, because it means there's a stiff rod down their back, but not necessarily a spine in all of them. So they get, and then this neural tube is for the most part a spinal cord. And even a tail, we start with the tail in early embryonic development. And like everything else that we've seen for the last few slides, we're bilaterally symmetrical, so just side to side. Now, if we delve further into those chordata, so you have to go within the Animalia group and then chordata as a subset. And now we look at some different classes that we need to know. We need to know fish. I think most of you could identify a fish from a picture. But we've got gills, scales, fins, and of course their fertilization is external in the water, and they're ectothermic. They don't regulate their body temperature. Amphibians, like our frogs and salamanders and things, some of which are called newts. So be careful that you identify that as an amphibian, not a reptile, these sort of guys here, because they have skin. They don't have scales. And most of them, they'll have like a tadpole stage as well. They often start little larvae in the water with gills, and mostly by the time they're adults, they breathe air with quite simple little lungs, and usually have forelimbs as well. But they're still ectothermic. They don't regulate their body temperature, and fertilization still takes place externally. And their eggs are not watertight, so they have to lay their eggs in water. Reptiles, on the other hand, can lay watertight eggs. So they're a bit leathery. They're quite different to bird eggs. But they're still ectothermic, still cold-blooded at this point. But they have a bit more developed lungs, and their skin is scaly or has overlapping scales. And they include things, of course, like crocodile, snake, and our tortoises here. A little chameleon. Then we get to birds. Birds can regulate their body temperature. They're endothermic. Their fertilization is internal, and their eggs are quite rigid. So you've seen a chicken before, I'm sure. They have feathers. They might be quite differently adapted for their purpose. Like the kiwi's little feathers are quite fluffy here, or like our penguin for warmth. And they, even though they've got wings, they won't necessarily fly. Like our penguins are adapted for swimming. And they live in sort of most habitats around the world. Birds have found a niche. And then ourselves, our mammals, which by definition have mammary glands. That's where we get the name, which is the ability of our females to produce milk for the young. And we also have hairs, sometimes not a lot. Whales only have very few hairs, but they do have them. And of course, well-developed lungs. We have 4 limbs, and most definitely regulate our body temperature and survive in all sorts of environment. Almost all mammals will have live young, with the exception of the monotremes, which is a platypus and echidna. Even marsupials have live young. They're just very small and go into the pouch. Now, with classification, you should be able to make some kind of key to be able to identify things or tell them apart. So you should be able to use the key or make your own or part of your own. Here's some examples. It could be like this kind of list of questions, and it will tell you which question to go to next until you get down to identifying species. Or this is the same one, just as kind of a branch, more visual, answering yes or no questions. So being dichotomous, di for 2, means there should only be 2 possible answers at each stage. So ideally a yes or no question. Sometimes they're not the best, the existing ones. Like this production of gas one, really, you're much better if you should be able to describe something that you can see in a picture. it's more practical for a field guide. So these kind of physiological processes aren't the best, but for bacteria, you can understand why they would use them. You want things that are objective. If you say something's really big, or if you say it looks nice, or you say that's skinny or something, or long, that's not very helpful. You should be able to quantify it so that someone else can use it regardless of their judgment. And you want things that perhaps, you don't have to dissect the organism, because most things in a field study, you'd be ethically obliged not to kill the organisms you're looking at. And you want to be able to look at even if the organism gets separated from its habitat. Sometimes you do see things like spiders, for example. A common thing in those keys are to look at their webs, but the strongest keys rely just on features of the animals or plants. So there's some examples there. All right, and that wraps up all our kind of key points from ecology and evolution. It's covered a lot in an hour. Please let me know if you have questions or if you want to go back over a particular component in more detail, but I hope that's helped at least kickstart your revision of those topics.